Hi everyone. So the paper I'm presenting today is called HiCLIP, which stands for Blood Cancer Learning through Omics and Ontology Aware Modeling with Hierarchical Clip. It's a paper from the Machine Learning for Genomics workshop at ICLR 2026 out of Duke University. Let's get into it. So what is this paper actually doing? At its core, HiCLIP is the first clip style contrastive learning framework built specifically for hematologic, meaning blood cancers. You may know Clip from OpenAI. It aligns the images and text into shared space. This paper does the same thing, but instead of images and text, it aligns like RNA derived data on one side, things like gene expression and fusion events, and DNA derived data on the other side, things like genomic mutations and variants. The goal is to push these two different views of a same patient's tumor into one shared embedding space. They call it as like lattice. Once you have the test, that shared space, you can do some really powerful things. You can impute missing modalities, classify cancer subtypes, and predict patient survival. Now, why did I choose this paper? There are few reasons. The first, it sits right at the intersection of three areas that are exploding right now: multimodal machine learning, biological foundation models, and clinical AI. Second, the technical contribution is genuinely elegant. Encoding a disease taxonomy directly into contrastive laws is not something that I had seen before, especially not at this scale. And the third, evolution is unusually rigorous. They explicitly test on held out hospitals and rare disease, which is rare in clinical machine learning. Here's a quick map about like what are we going to see today. There are 7 sections. We'll start with the problem: what makes blood cancer hard and why data is such a mess. Then I'll show you what existing methods get wrong, and then method itself, the results, my own critics, and we all like wrap up with the conclusion. All right, the problem: blood cancers affect more than 10 million people worldwide and account for around like 10% of all cancer cases globally. That's a huge number, but here's what makes them particularly nasty from a clinical and machine learning standpoints. Look at the number 165. That's how many distinct disease entities exist under a blood cancer umbrella, all organized in 5-level hierarchy defined by World Health Organization in 5th edition classification, and it's called as like WHO 5. So this isn't one disease, it's more like 165 related disease, each with slightly different biology, genetics, and treatment. On the left side of the slide, the clinical challenge, diagnosing these subtypes requires integrating pathology, immunophenotype, genetics, and clinical features simultaneously. And some subtypes are so rare that you might only find 50 patients worldwide. That makes training any supervised model extremely hard. On the right side, multi-omic profiling, combining RNA sequence with exome sequencing, has genuinely improved subtype classification and outcome prediction. So the tool exists. The question is how to use them when your data is incomplete, heterogeneous, and spread across 28 different hospitals with different protocols. That's exactly the gap this paper is talking about. As you can see from the Venn diagram, and this is a real data from their cohort, 34% of patients are missing either their real RNA or their DNA profiling. Only 65.9 have both. So more than third of your patients just vanished from a standard multi-omic model because they are excluded. That's not a small data quality issue. That's a fundamental problem with how clinical genomics works in practice. Sequencing is expensive and not everyone gets the full panel. The second challenge is out of distribution shift. They held out 3 entire hospitals. That gives 4 evolution settings. And the hardest one is IO-DEVO has new hospitals and rare diagnosis simultaneously. That's a close to real-world deployment, as you can get it in the paper. The third challenge, variable length event sets. One patient might have 2 somatic mutation, another might have 47. Standard neural network that takes a fixed dimensional input just can't handle this without losing information. So what does the field currently offer? MOFA Plus is probably the most well-known multi-omic integration method. It's a factor model that finds shared latent structure across modalities. Solid, but it can't handle a completely missing modality. It has no notion of disease hierarchy. Then you have a clip style methods like ECECLIP and ECEN. These are actually contrastive learning methods, but they are built for a single cell data, not a clinical bulk coherence, and no hierarchy awareness. Bulk RNA BERT is a foundation model for bulk RNA sequencing, genuinely powerful transcriptomics, but it is single modality, no DNA, no imputation. The nucleotide transformers DNA equivalent, great for variant sequence, but again, unimodal and no patient level aggregation strategy for variable length variants. And then you have hierarchical softmax classifier. These do use the disease taxonomy, but they are supervised classifiers only. They can't impute missing modalities and they don't produce transferable embeddings. So everything in the field solves at most one of the 3 problems, as we just identified. Nothing combines them. That's a real gap over here. This is the important table from the paper. Look at the columns: multi-omic, missing modality, OOD, robust hierarchy, and parameter efficiency. And look at all those redux. MOFF Plus handles multi-omic but not missing modalities and not hierarchy. ECECLIP handles multi-omic but nothing else. Bulk RNA BERT is partial on OOD robustness because it was pre-trained broadly, but it still only works on RNA. Every single method has a gap somewhere critical. Now look at bottom row, HiCLIP. Double check marks across the board. Multi-omic yes, Missing modularity, yes. OOD robust, yes. Hierarchy, yes. And it does this with a low parameter count because it freezes all the foundation model encoders. The bottom box on the slide captures it perfectly. No existing method simultaneously handles the entire modality missingness through principal imputation. OOD shift across institutions and rare diseases, and disease hierarchy consistent geometry. That's a gap, and Bloom-HiCLIP is the first method to close all three simultaneously. Let's look into proposed method. So how do they actually solve it? The core idea, and this is actually highlighted box at the top, is elegantly simple. Treat RNA and DNA as a two different views of the same tumor. If you are studying a patient's cancer, you could look at it through a RNA lens, what genes are being expressed, what fusions are happening, or through the DNA lens, what mutations are present, what genes are altered. Both views describe the same underlying biology, so pull them together with a contrastive learning, and you get a shared space where RNA and DNA embeddings of a same patient land near each other. And here's why that solves the missing data. If a patient is missing their DNA, you could use their RNA embedding to drive the nearest neighbor in the shared space. And then you use the neighbor's DNA embedding as the substitute. Imputation becomes a retrieval problem. That's beautifully clean. For OOD challenge, encoding the WHO5 taxonomy into the loss function means that patients with related diagnosis, even one from held out hospital, are pulled into biologically coherent neighborhoods. The hierarchy acts as a prayer that generalizes across institutions. For variable length events, they tokenize each mutation and fusion as sequence element, prepend a learnable CLS token, and run a transformer. The CLS token output is a patient's embedding, variable length in fixed dimension out. It's a two-tower clip model, just like original clip, but for genomics. On the left, RNA tower, it takes in four types of inputs: bulk gene expression via bulk RNA BERT, fusion peptide sequence via ESM2, that's a metaprotein for language model, gene set embodings via GSFM, and tabular RNA quality controller metadata. All these get projected and tokenized, then processed through a small transformer encoder. The CLS token output becomes RNA's patient's embedding. On the right side, the DNA tower, same idea, but for DNA. Variant nucleotide sequence through nucleotide transformer, cleanware disease term, text via like PubMed BERT, gene set embeddings again through GSFM, and tabular DNA quality control data. Both tower output a 512 dimensional embedding, L2 normalized. These two embeddings get published into lattice, the shared latent space in the middle, by the hierarchical contrastive laws. So I just walked you through the architecture using a diagram that was built, but here's the actual figure from the paper itself. It ties everything really clean. Start on the far left, you have the full ABCG coherent 8,281 tumors. From there, it fans out into four evolution splits, as we discussed. IIDI is your main training and test set, and the three OOD splits, IIDO, IODI, and IODO, are held out entirely for evolution. Cooling down the left, you can see the input data broken into two streams: multi-omics on the demographics, disease stage, family history, blood counts, and serum chemistry. The multi-omic data feeds into HiCLIP in the center, the RNA tower on the top, DNA tower below. Both towers output their respective embeddings, and those get aligned into lattice, that shared embedding space as shown in the column of the blue square in the middle. For hierarchical diagnosis, the same lattice embedding is combined with the diagnostic clinical encoder output. Note these two are two separate clinical encoders because the relevant clinical features differ between the two tasks, and the feeds into hierarchical softmax model at the bottom right, which produces a prediction along with WHO5 tree. The color tree at the very bottom represents the hierarchical prediction. Each model is a branching decision, and model can stop early at any level if it is not confident enough about the subtype. That's a whole pipeline end to end in one figure. Okay, let us look into the hierarchical loss. Standard info NCE, which is what CLIP uses, treats every non-matching pair as equally negative. Look at the matrix on the left. In a batch like AML, CML, follicular lymphoma, and DLCL, AML and CML gets a weight 0 between them. Same as AML and DLCBL, but biologically AML and CML are both myeloid cancers. They share a cell of origin and enormous genomic overlap. Treating them as equally negative is just wrong. It forces the model to learn an embedding space that ignores real biology. Now look at the right side, the HiCLIP soft weights. When two patients share the same diagnosis, weight is 1.20. That is your true match. But when two patients share the same cell of origin, like AML and CML, both being myeloid, and weight is 0.26, not 0, but not a perfect match either. Patients from completely different lineages, like a myeloid and B cell cancer, get 0.07. We are not forcing those apart completely. We are just like deprioritizing them. The key insight is, instead of binary targets, match or no match, we have soft targets derived from the WHO hierarchy. The loss function literally encodes biological knowledge about the disease relatedness. So the result is plus 52% improvement in diagnosis level hit rate over standard N4 NCE. The hierarchy is real, the biology is real, and encoding it helps enormously. Two more pieces before we get into results. Tokenization strategy on the left solves the variable length problem. You start with CLS token, learnable patient level summary, then you add fixed tokens for a bulk expression. and then tabular features. Then you add event tokens for each fusion or mutation, up to 32 events per patient, kept in order of coverage. Shorter sequence, just add PAD tokens that are masked during attention. The transformer sees the whole sequence and CLS token aggregates it all. Kind of clean and scalable. On the right, the parameter efficiency, Bloom and HiCLIP only trains 39.5 million parameters compared to that 121 million if you fine-tune the smallest version of the each foundation encoder, or 1.32 billion if it went full fine-tuning. That's 33.4 times fewer parameters, and it still outperforms. Freezing the encoders is not just cheaper, but it also better regularization, which helps the OOD generalization. The dataset is ABCG, 8,281 tumors, 165 subtypes, 28 institutions, all with expert panel review diagnosis. That's genuinely the largest multi-omic blood cancer cohort to date. They split this into 4 evaluation cohorts based on 2 axes: institution identity and diagnosis identity. IIDI is in distribution on both your standard test set. IIDO tests generalization to rare disease. IODA tests generalization to new hospitals. IODO is the beast, new hospitals, and rare diagnosis simultaneously. That's the hardest possible real-world setting, and they report results on all 4. The results are strong, starting with retrieval. 94.8% recall at 5 on the test set. That means when you take a patient's RNA embedding and try to retrieve the DNA counterpart from the database of thousands of patients, the correct match is on the top 5 candidate, almost 90% of the time. But here's what I find more impressive. The IODO results. Remember, IODO is the hardest setting. New hospital, rare disease. Recall at 5 is like still 92.9% recall. At 1 is 74.9%. I mean, reciprocal rank is 0.828 and completely held out. Said with the disease the model has never seen from hospitals. It will have never trained on. That's a robust generalization. On the right, imputation quality. Top 1 quotient similarity of 0.82 on the test set means imputed embedding is geometrically very close to the true one, and it degrades gracefully, even on IODO. But we are still at 0.71. That's a great thing. For a hierarchical diagnosis, they built a hierarchical softmax model on the top of the lattice embeddings. The heading number is like 98.1% micro averaged for the cell of origin classification. That means the model is excellent at deciding whether the patient cancer is a B cell, myeloid, T cell, NK cell, or histocytic origin. The granular subtype classification across 29 subtypes, which is genuinely hard. Test set top 1 accuracy is 34. That sounds low, but the task is classifying into 29 finely defined categories with sometimes very small training sets. More importantly, look at the hierarchical distance, 2.0 on the test set. That means when the model is wrong, it only wrong by about 2 edges in WHO tree. It is confusing AML with APL, not AML with DLBCL. Those are local clinically interpretable errors. And critically, on IODO, the hardest split, accuracy is 15.5%, which is the best of all baselines. The model holds up even when facing hospitals and disease is never seen. The adaptative early stopping also means that model says, I'm not sure about the exact subtype, but it is definitely acute myeloid leukemia, like rather making a confident wrong call. For survival prediction using a deep hit, overall survival C index is 0.788 on validation. For a context, a C index of 0.5 is random and 1 is perfect. 0.78 is solid clinical performance. And look at the IODO OS index 0.698. Even on the hardest OOD split, the model maintains a meaningful prognostic discrimination, I would say. The freezing encoders is just not cheaper. It likely acts as a regularization against overfitting to a training hospitals, and forced survival again over clinical only is real, but modest. Genomics complements clinical features like age and CBC, but it doesn't replace them. Let's talk about the critics. The most important gap labeled high severity here is the missing comparison to established multi-omic integration methods. MOFA is the standard baseline in this space, and it's absent. Even more pointed, what if you just concatenated all 5 frozen foundation model embedding and trained a supervised head? Would that match or beat Bloom HiCLIP? We simply don't know. Without the baseline, we can't isolate how much of the gain comes from CLIP objective versus just having a rich pre-trained representations. And the second, 34% top 1 accuracy is hard to interpret in isolation. The hierarchical early stopping mechanism means like close but not exact counts favorably in the distance metrics. I would want to see separately reported like what fraction got the exact subtype right and what fraction got at least the correct cell of origin. The framing of like errors or local is reassuring but potentially like a flat wing. And the third, survival improvements over clinical only are marginal and crucial. They don't formally test whether the C index difference is statistically significant. That's a gap. And the fourth, interpretably, like lattice is a black box. We can retrieve similar patient but can't explain which mutation or fusion drove the similarity. That's a real barrier to a clinical adoption. And the fifth, I would say, they test OOD on different hospitals, not different time periods. Concept drift as like WHO classification evolves in a deployment reality, which they haven't addressed it. Let's talk about the contribution and the future directions. The key contribution C1, uh, first hierarchical multi-omic clip for blood cancer. The WHO5 taxonomy is baked directly into geometry of the embedding space. And C2, soft target contrastive loss, encoding taxonomic distance via exponential decay weights. That's the technical heart of the work. C3, CLS pooling tokenization that elegantly handles variable length molecular event sets. C4, parameter efficiency, 33.4 times fewer trained parameters than fine tuning by freezing all 5 foundation encoders. C5, transfer learning that actually works, like 98.1% COO or for diagnosis and C index for 0.788 for survival. The most compelling future direction in my view are three: adding copy number variation and histopathology images as additional modalities, developing attribution methods so clinicians can understand like what is driving patient similarity, and extending this framework to treatment response prediction, which is arguably the highest valuable clinical downstream task, I would say. Here is a summary at one place: 4 tasks, all strong, 94.8% retrieval recall at 5, cosine similarity 0.824, imputation 98.1%, aura for cell origin classification, C index of 0.788 for overall survival. And all these are significantly better than a single modality baseline across all 4 evolution cohorts. The results holds up everything that they have tested. Blood cancer is hard, heterogeneous clinical problem. Existing methods can solve one-piece puzzle. Bloom HiCLIP is the first to close all 3 gaps: missing modalities, OOD, shift, and hierarchy simultaneously in one parameter efficient architecture. The results are strong and they hold up on held out institution and rare disease. The critics are real, but the contributions are really substantial. And that's all about Bloom HiCLIP. Thank you very much. If you have any questions, please post.